Dr. A.P. Abraham, MD on Pathways and Medicine. We joined in 1987, and after we were doing MD together, or at least the head of hematology department, the professor head of hematology. I was explaining to MD that before COVID started, we decided to have monthly lectures for faculty and PGs on newer advances so that in medicine as faculty we can keep ourselves updated. That was the idea of having these lectures. During COVID, it all went completely online. That was another experience. But now that COVID is sort of over, or may not ever be over, we have decided to go back to offline classes so we can also meet each other and meet the faculty who are taking lectures for us. So I'm very grateful to everyone who agreed to take this lecture for us on heart disease. I'm not sure, some time ago we thought stem cell was the magic panacea for all conditions, all cancers. Now we are reading CAR T cells as being some sort of a magic for all cancers, the ultimate sort of magic bullet. So we just wanted to have some idea about what are CAR T cells and what is the current state of science in that. I want to thank Jennifer who organized this meeting and sent out notices and invited this year. Thank you, Jennifer. So without much ado, I'll ask Gary to start. Thank you.
Good morning, everybody. Thank you, Tabu, for giving me the opportunity to talk. So, like what Tabu mentioned, so it's interesting how CAR T cell evolved. I'll kind of briefly go through the history of how it all happened. And I won't restrict myself to just CAR T cells because the entire thing is intertwined. The cell therapy per se is intertwined. And like he mentioned, stem cell transplant was the first, I will say, that in itself is a cell therapy. It's not just, you are transferring the stem cells to another person and that brings up the entire cell spectrum in the hematological as well as immunological milieu. So, the immune system in the body is constantly on the vigil. So, there are many similarities when it comes to infection and tumor. Many patients who come to us, I face the question frequently, doctor, how did cancer develop? How did this develop? And the simplest explanation I can give is based on the immune system. So infection is foreign in origin and it comes from outside the body, but it could be part of the body, arise from the body flora itself, the infection. But it's always recognized as outside, coming from outside. And there is always a constant interaction of the microbes and the body's immune system. And when the balance breaks down, there is infection. And if at all that occurs due to any cause, the balance is irreversibly in favor of the infection. Compare that with the tumor. Again, the difference is stark. It's endogenous in origin, it's not foreign in origin. And it has the Trojan behavior. And that helps it survive. Unlike the infection which comes from outside and recognized as coming from outside, the Trojan behavior of the tumor is what helps it survive. And continuous cell damage due to various physical and chemical agents and generation of rogue cells that perpetuates cancer. And here again, there is a constant interaction with the immune system. And when the balance breaks down, it becomes cancer. So, cancer doesn't develop overnight. It's a matter of many years. And elegant studies have shown, as you follow patients over a period of decades, as they advance in their life, cancer and aging is very closely interrelated. And the incidence of cancer goes up as the age advances. And why does this happen? It's because the body's immune system is not able to cope up with the development of these cells which become cancer. So we have this myelodysplastic syndrome which graded and going all the way to AML. That's a classic example of how cancer develops for a period of years. And when it comes to death, there are no winners. The patient dies and the tumor also dies, unlike the infection. Next slide. So, how do you approach the treatment in infection and tumor? So, you can prevent infection through vaccine, avoid exposure to the infectious agent. You can use chemicals, which are the antibiotics that are of course immune therapies as well. You can use GCSF to push up the counts, randomized infusion, which we use, antibodies, even cell therapy. I will briefly mention that later. You can use cells also to fight infection. And you can use surgical methods for incision and drainage of an abscess. So compare that with the tumor. You could avoid high-risk behavior to prevent cancer, environmental modifications. You can use chemicals again, which is called chemotherapy, surgical resection or radiotherapy. And again, you can utilize the immune system to treat cancer. Monoclonal antibody, immune modulators, other agents, and cell therapy. The entire spectrum is there. So again, that's quite similar. So this picture actually shows the tumor microenvironment. So for the tumor to grow, it needs nutrition and oxygen, of course. That comes through the blood. So supply of blood or nutrients is very vital. And as the tumor grows, just like ecosystem, full ecosystem, in the forest there are many kind of trees and like that, there's an entire ecosystem which is there supporting the tumor. And the tumor manipulates the entire ecosystem for its benefit. That's how it thrives. And the immune system become either passive or active players in helping the tumor grow. So here you can see that there are tumor, the dendritic cell, activated dendritic cell, tumor associated macrophages, and myeloid-derived dendritic cell. So here you can see the dendritic cell, myeloid-derived stromal cells, and the tumor-associated macrophage are important players in the tumor microenvironment. And the cytokines produced from tumor-associated macrophages stimulate the T cells, and as well as the dendritic cell, which presents the tumor antigen through MHC class to the T cell, activates the T cells, and it generates this signal too, and it could potentially attack the tumor. And then there are danger signals which are produced, and all these things will amplify the inflammation. And also, when you look at this, you could potentially kind of modulate any of this area for our benefit so that you can kill the tumor. So from this you can understand that modulating the immune system, either through the manipulating the T cells, the macrophages, myeloid-derived stromal cells, dendritic cell, any of this, you can actually potentially treat. These are all potential drug or any other target by which you can treat cancer. Next one. So this is in a nutshell of the, I'm sorry if it's not very clear, the T cells are stimulatory and inhibitory signals. So T cell receptor is a complex receptor, and once for it to bind to an antigen bearing cell, it needs MHC class molecules. On the left side you can see that MHC class and the antigen is presented by the MHC class molecules, and the T cell receptor is docked there. And there are many co-stimulatory molecules here, CD28, 1BB, CD40, like that. There are many of those on the left, on that side. And here there are many inhibitory signals, which include CTLA4, PD1, and there are many other inhibitory signals. So why I put up this is the mere docking of T cell receptor to HLA class antigen is not enough for it to act. It needs many co-stimulatory and co-inhibitory signals. Why do you need co-inhibitory signal? It's a peripheral, to induce peripheral tolerance. Otherwise T cells will become autoimmunity. That's what happens. It goes on, it's a loss of peripheral tolerance which lead to autoimmunity. So that's body's way of checking it. But these are all again potential targets for immunotherapy in cancer. Next one. So this Nobel Prize in 2018 was awarded to these two researchers for discovering the inhibitory part of the T cells. So Dr. Alaizan from Texas and Dr. Honjo from Japan. So Dr. Alaizan discovered the CTLA4 and Honjo discovered the PD1. These are two most important co-inhibitory signals in the T cell interaction with the other cells. Next one. So having said this, I briefly mentioned there are different modes of immunotherapy in oncology possible so far. You could use monoclonal antibody. I'm sure all of you would have used rituximab in autoimmune conditions. We use rituximab like water. We deal with diffuse large B-cell lymphoma which expresses CD20 and therefore it's and the rituximab had made a revolutionary change in the way B-cell lymphomas are treated. Inotuzumab, it targets CD22. And again, it works the same way as rituximab. The only thing is that it has got is conjugated to calciumycin, which can cause cell damage. Tubular damage is like vincristine. But it's a monoclonal antibody. There are bispecific antibodies like blinatumomab. It works like what I mentioned earlier. The way it works is it binds to T cells on one side, and it also binds to an antigen of interest in the target cell. So it brings the T cell to the target. So the T cell may not directly attack the antigen of interest to us, but this antibody binds on both sides and makes the T cells attack the tumor. That's what blinatumomab does. And the target is CD90, which is very much expressed in lymphoblastic leukemia cells, lymphoblastic leukemia cells. So blinatumomab is a drug which has revolutionized treatment of ALL. Then there are immune checkpoint inhibitors, nivolumab. So that acts on the PD-1, PDL interaction. Again, like what I mentioned, it removes that inhibitory influence, that co-inhibitory stimulus. So because of that, there is another drug called pembrolizumab. It acts on the PDL1. So either way, through immune checkpoint inhibitors or bringing the T cell closer to the antigen and increasing its effectivity, you can effect many tumor kill as you wish. Of course, stem cell transplant, you are actually giving the entire new immune system from the donor to the patient, so that you, with the expectation that the immune system you are transplanting will take care of the tumor. Donor lymphocyte infusion, which is mostly T cells. That's a tool we use when we see post-stem cell transplant. If the donor cell count is reducing, we infuse donor lymphocyte infusion so that the immune system is upregulated and it brings up the cells. So when we transplant stem cell, we are not just transplanting blood, we are not just transplanting the myeloid cell, we are transplanting the immune system also. We are giving blood as well as the immune system. So in non-malignant conditions, we use DLA to bring up the donor status. The donor cell count should come up. What we call chimerism, whereas in malignant condition, especially CML post-transplant, we use this to control the disease. If we feel that the disease is bouncing back, we can use a donor lymphocyte infusion. Then there is in vitro culture cells. So donor lymphocyte infusion is taken directly by ferricing the donor, whereas in vitro culture cells are used by, you can use either the patient cell or the donor cell, culture it in vitro and infuse it back. That is called adoptive cell therapy or ACT. And CAR T cell is one part of adoptive cell therapy. And CAR T cells is the modified in vitro culture cells. So how can you do adoptive cell therapy? There are both innate immune cells and adaptive immune cells. And it's not just T cells which can be utilized for adaptive cell therapy. You can use NK cells, gamma delta T cells. Gamma delta T cells don't express, they don't recognize HLA based antigen, they don't need HLA, MHC. And adaptive immune cells include tumor infiltrating lymphocytes, TCR T cells and CAR T cells. TCR T cells are nothing but T cells whose T cell receptor has not been modified. Whereas CAR T cells have a receptor which is modified, which is a chimeric receptor, chimeric antigen. That's why it's called chimeric antigen receptor. So, how did this all start? Tambu mentioned that he ended medical school in '87. So, the same year this happened. So, it's a long story. So, again, Dr. Yoshihisa Kuwana from Japan and to Dr. Gross and Eshar from Weizmann Institute of Science in Israel. They made the first chimeric antigen receptor. It's a crude thing, like anything, anything which is made first, it won't be that fantastical, but it all started with that. So they call it T-bodies. the T cells with that kind of chimeric receptor. They call it T bodies. And the first CAR T cell therapy happened in 2012. And this girl, in 2012, her name is Whitehead, Emily Whitehead. And this is a picture from the foundation. So she's the first girl to get, for a relapsed ALL. Her parents were advised stem cell transplant. They did not want their child to undergo stem cell transplant because they thought there was a lot of complications. And I agree that in ALL, transplant in a relapsed ALL don't produce very good outcome. That's one area where probably drug therapy is possible. But until that time, nobody had used CAR T cells. They decided to use, and this girl is cancer-free 10 years. This is a picture this year. So this happened in 2012. So from 1987 till 2012, what happened? The T-bodies were discovered in 1987. There was painstaking work involved. So just because you produce something doesn't mean that it will produce good clinical outcome. It took 25 years of painstaking, slow progress to reach this milestone. Next one. So this picture you can see the T cell receptor and the chimeric antigen receptor. The MHC class and molecule presents a tumor antigen to the T cell. You can see the T cell receptor which has got alpha, beta, epsilon and psi, gamma, and alpha, beta, delta, gamma and psi substructures to it. And here you can see the chimeric antigen receptor. Like I mentioned, why is it called a chimeric antigen receptor? Because it has got parts of a T cell receptor and it also has a part of a immunoglobulin molecule. Though this is the immunoglobulin part of it. So short chain of variable segment, the short chain is also part of the receptor. So we call chimerism when the patient has got a stem cell transplant. We check for how much of donor, what proportion of it is stem cell donor cells and what proportion is the patient's own cells. We say 100% chimerism means 100% donor cells. In a similar way, the chimera is a Greek god who had half animal, multiple animal or whatever. So that's why the name has come. So here there is a hinge region, there is an extracellular domain, there is a transmembrane domain and an intracellular domain. And the extracellular domain is the chimeric part. Subsequently, the transmembrane domain, then there is a co-stimulatory domain. One or two co-stimulatory domains are there. The advantage of this receptor is that it can bind to the tumor antigen without the HLA or MHC class. That is very important because it doesn't need the body's own immune system to help it. It can go and directly attack the antigen of interest. Next one. So, like I mentioned earlier, you can engineer T cells without modifying its receptor into a chimeric receptor. So they are called engineered TCR T cells to differentiate from CAR T cells. And somehow the development of engineered TCR T cells has not kept pace with the CAR T cells because obviously there are many limitations. Here you can see, you can compare, you see the comparison of the receptor which I showed earlier. So the function, the TCR T cells function through well understood T signaling pathways. It does not, it cannot work without MHC antigen. So it is MHC restricted. And it cannot target non-protein surface molecules. Those are the disadvantages of TCR T cells. CAR T cells can recognize tumor cells independent of MHC. It can target non-protein surface molecules for like carbohydrates and lipids. But it only recognizes antigens expressed on the cell surface. Which is, it can, even though it can recognize which are not, which is not expressed by, which is not presented by MHC, it cannot go inside the cell and kill it. It's only the outside the cell. Next one. So CAR T cells, there is a signal amplification from the synthetic biology. So even just 200 targets can trigger a CAR T cell. Ability is controllable. It can target surface molecule structures like proteins and glycans. MHC independent. It could last for a decade at least, and they are serial killers of tumor cells. That's a typical word used, serial killers. And cytokine release syndrome is more severe than with T-cell based therapy. We will come to that. It's a very important part of T-cell therapy, how the cytokine release syndrome happens. So T-cell engineered T-cells, the sensitive signal amplification is derived by evolution of the receptor. It has got low avidity. That explains how it is not kept pace with the CAR T-cells. It targets intracellular proteome, TCR, requires MHC class 1 expression, lifelong persistent serial killers of tumor cells though, and the off-tumor toxicity is difficult to predict in the TCR engineered cases. Next one. So, how has CAR T-cell receptor or CAR T-cell modification evolved over a period of years? It has gone into the 4th generation and 5th generation. I'll come to that. The 1st generation CAR had that the extracellular domain like that, and it had a His region and a co-stimulatory molecule. And the 2nd generation had an additional co-stimulatory molecule, which is CD28 or 41BB. I had mentioned this when I mentioned about the co-inhibitory and co-stimulatory T-cell, the molecules, co-stimulatory and co-inhibitory molecules of the T-cells. Then 3rd generation added both. There are 2 co-stimulatory molecules in the intracellular domain. Next one. So, what are the modifications of CAR T-cells? It increases the potency, CAR structure, gene editing to remove. So, you can potentially modify the CAR T-cells further. You can use gene editing, DNA editing or RNA editing to prevent the expression of certain molecules. So, you can make the CAR T-cells kind of not express the ligands for CTLA for PD-1. So that means that it will not be coded down by the CTLA for PD-1. It will kind of inhibit the regular T-cell. We can increase the specificity. We can use 2 separate chimeric antigen receptors in the same CAR T-cell, or we can use 2 separate CAR T-cells which target different antigens. And basically, like any microbes, a word, therapy by developing resistance. Tumor cells also can develop resistance to any therapy which we use. We have tumor malignancy, cancer resistant to regular chemotherapy. They can develop resistance to CAR T cells. So the idea of combination chemotherapy in TB or any other infection holds true here also. You have to probably use 2 targets, either in the same CAR T cell or use 2 different CAR T cells. So you can increase the safety. The safety can be, so CAR T cells can produce many undesirable side effects. You can incorporate a suicide switch, and if you want, when you want to switch off the CAR T cells, you can utilize that suicide switch. An example is the thymidylate kinase based system, where you put that inside as part of the CAR T cell construct, and then you give ganciclovir. The entire T cells will get killed. So that can happen. There are many other ways of doing it. I just gave one example. You can reduce the cost. So CAR T cell is made from autologous T cells. You use the patient's own cells. Why? Because otherwise the patient can have a graft versus host disease. The donor cells can potentially attack the patient. So, and therefore we usually use the patient's own cells, and that increases the cost because it is basically it is made for that patient. The entire process is. So what is the advantage of large pharma-driven therapies which we use day in and day out? So there is, for example, paracetamol. It can be used in anybody. So it can be mass manufactured. But CAR T cell is made only for that patient. So the cost is phenomenal. So one way of cutting down on the cost is to use universal donors. There are ways of doing that. There are HLA antigens which are commonly expressed in the population. You could catch one of those people and use their cells, and therefore the body doesn't, their cells don't mount a response against the patient cells. So the universal off-the-shelf product and you could also, in addition to that, the other way of doing that is use gene editing tool. Like I mentioned, CRISPR-Cas9 system, which can silence any gene. Use that system to silence, to make the HLA expression, to remove the HLA expression. Next one. So this one actually uses the CAR-T, there are expression of two CAR receptors through notch, through a mechanism through notch. So here you have made a car, and that car is actually going and making another car. So that is one way of doing that. Like I mentioned earlier, a CAR T cells can be engineered to express two chimeric antigen receptor. This is one way of doing that. Next one. So people are moved from car to, I just mentioned armored car, it's not literally, armored car means with this modification you can make the CAR T cells more powerful. So CD40L, it stimulates a potent anti-tumor response by binding to CD40 and nearby B cells, macrophages or dendritic cells. So you can actually make the CAR cells express CD40L to bind the neighboring immune cells which we saw in the tumor microenvironment loitering around, and make the CAR T cells more effective. IL-18, many of the cytokines are important in immune cell activation. So you can make the CAR T cell express IL-18, which will increase the activity of NK and CD8 T cells and interferon gamma secretion, and macrophages and antigen presenting dendritic cell will become much more active. So this second thing is also called TRUC. Basically, using the cytokine, the T cell receptor modification using cytokine. So that's another short form called TRUC. Then you can use PD-1 blocking molecules, so effective for blocking the PD-1 receptors. So these are the different ways of making the CAR T cells more effective. Next one. So what I mentioned earlier, you can see here CD40L CARs, IL-18 secreting CARs, PD-1 blocking CARs. So these are the three different mechanisms through which you can modify the CARs and make armored CAR or TRUC, whatever. So now, having explained all these things, what are the limitations of CAR cells? It is a magic bullet, but it's not a panacea for all and everything. It cannot do everything, and it has got its own limitations. What are the limitations? There is antigen escape. Like I mentioned earlier, resistance can develop. So, one example of targeting this is in ALL we target CD90. Emily Whitehead had a CAR T cell which targeted CD90. Inotuzumab targets CD22. So you can make a CAR cell which will target both CD19 or CD22, or use two CARs which will target CD19 and CD22. Solid tumors, you can use the same thing for targeting glioblastoma, breast cancer. So single versus multiple targets. Then there is something called on-target off-tumor effect. Basically, the CAR T cells attacks the tumor, but it also goes and attaches off target and creates destruction there. So how to prevent that? You can actually target the post-translational modification of the tumors, which is specific for tumor cells, not for the other normal cells. So that's one way of doing that. Then CAR T cell trafficking and tumor infiltration by, you can administer the CAR T cell locally directly into a solid tumor, or make the CAR more effective, which can penetrate through the physical barriers. There are many ways of doing that. Like you can express certain enzymes which will break down the physical barrier. Immunosuppressive microenvironment, you can use immune checkpoint inhibitor in combination with CAR T cells. Therefore, that tumor microenvironment is modified. Then to reduce CAR T cell associated toxicity, I mentioned earlier the suicide switches, that is a CAR off switches. Next one. So in a nutshell, many potential targets are there, but overall of the story so far, the ones which are commonly used is ALL, CD19 and CD22 targets, myeloma, B cell membrane antigen, and in lymphoma, CD22, as well as CD13 Hodgkin lymphoma. In B cell lymphoma, CD22, and in Hodgkin lymphoma, CD13. These are the targets for which it has been used and found to be effective, clinically relevant effectivity. Next one. So this picture shows how it is all done. So you have to have the patient connected to the apheresis machine, which works like a dialysis machine. I'm sure all of you would have seen the apheresis machine. And the leukapheresis is done. the cell antibody or the cells are collected. There are many different ways of, in the next slide I will explain that. There are many ways of separating out the cells, and then you use the transduction. You have to have a vector construct. So it can be a viral construct, or you can use other techniques. So you have to have that chimeric antigen gene, which is and that has to go into the T cell. Then only that chimeric antigen receptor will be expressed by the T cell, and that's making that construct is very crucial. And that's the kind of the technology which takes a lot of time to master, and it's not easily available. Once you have that, you can manufacture the cell anywhere. You can do anywhere. There are platforms which can do it for you. And then you can see the G-Rex class where it is grown. Then it is expressed on the CAR T cell expansion, and then it goes back to after chemotherapy to the patient. So this is a picture from the Tisangen cell. So screening, leukapheresis, bridging chemotherapy to the patient if required, then the lymphodepleting chemotherapy. We use lymphodepleting chemotherapy to the patient so that the CAR T cells grow. Otherwise, because body's own immune system is working, means it could kind of potentially eliminate the CAR T cells which are infusing. So you have to use lymphodepleting chemotherapy, then the Tisangen Declusel was infused and then follow up. That's what happened. Next one. So this picture again also shows almost the same thing. It's a little more detailed, major steps in the CAR T cell manufacture. So essentially you have to use apheresis. So apheresis product you're collecting on the left side. Patient, you do apheresis. You can use many apheresis machines, any of the apheresis machines. We use the COB, now we have moved beyond COB. So you can use any of that. There is a system called Prodigy written there. We are using currently Prodigy system. We have not started that, but we are on the way. So we are going to use the Prodigy platform, and then the Clinimax Prodigy platform. You can use that once the cell is apheresed, you can use the magnetic bead system or any of the other technique to kind of separate the cells. Then T cell selection is done. Then using different techniques, and you can then you activate the T cell with either beads or expander or whatever, and then you use the gene transfer. For that you need a retroviral or lentiviral construct, which I mentioned. So retroviral and lentiviral has certain advantages. They are very efficient, but there is always a concern that it could potentially cause tumor in future. Because they are retroviral more so. And we could use a technique called electroporation to avoid the use of virus. Electroporation is nothing but you put the cells in a culture medium, you pass an electric current through it, the cell surface becomes permeable to DNA and all the other molecules. So RNA, DNA can get into inside the cell. That's called electroporation. So you put the cells suspended in a medium, liquid medium, you add the DNA or whatever thing you want to go into that cell, and then transfer high frequency, high volt, I can't remember exactly, and these things get inside. So that's called transposon. You can even make mRNA get into the cell. That's called mRNA electroporation. Then you do once that is done, that gene transfer is done, you do the T cell expansion, G-Rex class or Prodigy system or whatever. Then you collect those T cells and wash it and the control. Then before freezing, you have to run it through certain product quality check. Then freeze it, you can use it, or you can straight away infuse into the patient. Next one. So these are the different tests we do before the releasing the CAR T cells for clinical use. So there are several parameters: safety, purity, identity, potency, and potency. So release criteria for each of these particular things is important. So first thing is about it mentions about the release testing for lentiviral and the retroviral CAR T cells. So retroviral and lentiviral CARs, like I mentioned about when I mentioned about paracetamol, manufactured in factories, you need large scale investment and you need large scale facilities which can manufacture cells in large quantities. Only in such facilities there you can do this. Whereas the second one, the transversion or electroporation technique is easier and it is safer in certain ways, like the copies of transient insertion are kind of limited, and purity, there are purity when it comes to, there are checks for all these things. Next one. So this is a Prodigy system. This is a completely it's an automated system. It's completely closed. You infuse the whatever the cells you want after apheresis, and also all the reagents what you want, and you will get the product in your hand. We have such a platform, and we are developing the CAR T cell. We are trying to kind of develop the CAR T cell once our and everything is through. So we don't have a construct. So what we are planning is we will probably buy a construct from to develop our own construct. It takes a lot of money and a lot of time and effort. In India, IIT Bombay, in collaboration with Tata Memorial Hospital, Bombay, has made the first construct in the country 2 years ago, I think, 2 or 3 years ago. They had a lot of funding, and there was somebody dedicated to doing that. And mind you, this is only for one target. CD90 means you won't be able to use the same construct for CD22. And it's a very, very dedicated system. That is possible if you have the volumes, or you have unlimited money. So there are different business models which we can use. So our idea is to probably buy a construct from somebody and do the rest of the things we could potentially use. Next one. So now, what are the trials which are being conducted in primary cancer receptor T cells? So here you can see the different reported trials which are used for the CAR T cells. The product is mentioned, the co-stimulatory domain is mentioned. The first one is they use in B-cell lymphomas. It was the co-stimulatory domain of CD28. The second one was CLL 41BB domain. The next one is children with ALL 41BB. So ALL, DLBCL, mantle cell lymphoma. Here again CLL, DLBCL, follicular lymphoma in Waldenstrom's, DLBCL and DLBCL. And the response you can see, well, the overall response is fantastic. Complete remission ranging from 20 to 60%. You might think that these figures are not impressive enough, but for a patient with cancer who have been multiply treated and there is no hope, this is fantastic results. 60% complete response means it's like heaven. So, and the problem is cytokine release syndrome. With all these things, cytokine release syndrome is a problem. Like the cytokine release syndrome was less of an issue with these certain products, which probably was targeted to engineer to produce less of side effects. Next one. This again shows the same thing. Again in lymphoid malignancy. So the target is CD22 in ALL in the first one, second one BCMA in multiple myeloma, the third one also the same, CD30 in Hodgkin lymphoma, the last one. The last one is CD90, CD22 dual targeting, DLBCL and ALL. Here again you can see very good responses, complete responses, and the cytokine release syndrome in certain Hodgkin is low because the disease per se itself is not that aggressive, but you can see that even in others, certain CAR T cells have less of cytokine release syndrome at all. Next one. This one, yes. This picture, this is from the last one. So Enigem actually just kind of puts everything together and mentions the response rate in each of these trials. So how far have we reached with regard to leukemia? The response rate is 83 to 93, and that is in adults. In children, 68 to 90. In CLL, between 57 to 71. Diffuse large B-cell lymphoma, 60 to 86%. Follicular lymphoma, 70. And multiple myeloma, it's a kind of wide range. And solid tumors have been lagging behind in this. Why? Because CAR T cells are not able to infiltrate inside the tumor. The tumor microenvironment is not able to kind of penetrate and act on that. Unlike liquid malignancy like leukemias and lymphomas. Lymphomas come in between sort of. Next one. So let's a little bit about the toxicity. So any drug will have a toxicity if it is acting. If it is action, there will be a toxicity. So cytokine release syndrome resembles systemic inflammatory response syndrome, and it can also cause CNS problem. The cytokine release encephalopathy syndrome or immune cell associated neurological syndrome (ICANS). That's a term for it. The current term is ICANS. The frequency can vary from 10 to 50% of these side effects. Next one. So how does the cytokine release syndrome cause a neurotoxicity? There is an alteration of the blood-brain barrier, and therefore there is all the cytokines seeps into the CNS, and it could potentially cause anything from delirium to icy bleed. Hemodynamic instability, capillary leak syndrome, hypertension, organ dysfunction, the liver or any other. So essentially cardiovascular, CNS and liver, these are the usual sites. Next one. Here it enumerates again the different systems and the different the side effects affecting the but major points I mentioned the previous slide. Next one. So these are the grade. There are different grading of this. In fact, as therapy advances, there are newer and newer ways of grading. So the first one is the consensus group. Then there is a University of Pennsylvania guideline, Memorial Sloan Kettering. Then CTOX working group. Essentially, they are graded based on how the patient's clinical situation is. Grade 3-4 means it's life-threatening. You have to act with alacrity. Next one. It's again about the toxicity product. Next one. So how do you tackle this? The important thing is to recognize this early. Initial part of it you can treat symptomatically, very mild ones, like infusion of more fluids. And if it doesn't respond to that, you could use tocilizumab. If it doesn't work, tocilizumab works through IL-6, right? So it's the same story. Yeah, I think in rheumatology, you people must be using tocilizumab. In COVID, there are also Anakinra, and the more severe ones will require dexamethasone. So essentially, that's it. So the important thing is to recognize this early and act early. Next one. So let's come back to the full. It has come full circle. The CAR T cell therapy. So is CAR the endpoint? No, certainly no. CARs are magic bullets, but it's not a panacea for everything. And after CAR T cell therapy, the data has shown that, or long-term follow-up, 10 years and more, it has shown that those people who underwent stem cell transplant following a CAR T cell therapy has the maximum survival. So it might work in certain patients without any further intervention, but ultimately you have to top it with a stem cell transplant. So the entire immune system that has to come and fight the tumor, and it's a fight till the patient's end of his or her life. It is a constant struggle. I'm sure this immune system working against a tumor is happening throughout our lives, whether it's a patient or a otherwise normal individual. That's what preventing the tumor from establishing. So this particular slide shows the lag behind of the rest of the world. This is about the CAR trials on the other side and the other all clinical trials worldwide. So you can see that the West obviously leads because they have the money and the time and technology to advance. Next one. So this one shows about the TCR results. I will not run through. Next one. Okay. Now briefly about other adoptive cell therapy. I've covered my CAR T cell part. This is important even more for you because you people will be dealing with infections. This particular graph shows post stem cell transplant what happens to the immune system. There is conditioning, and we use a condition so that the donor cells are not rejected by the body, even though we do HLA matching. The nephrologist and solid organ transplant physician use HLA cross matching. We use HLA matching. Why? Because we are transplanting the immune system also. So we need HLA matching. For them, HLA matching is not required. There should not be a cross reaction. They are not changing the immune system. Solid tumor transplant physician don't change the immune system. We are changing the immune system. Therefore, the HLA has to be matched. And here you can see that if you use the conditioning, the body's immune system is really downgraded. That is day 0 when the stem cell is infused. And you can see by day 14, the cells will slowly engraftment happens. The first things to recover will be the neutrophils, NK cells and monocytes. CD8 T cells and B cells might recover by around day 100. And the CD4 T cells might recover 12 months or even 2 years to recover. So during this time, lot of infections can take place. You can see that. So it can be a bacterial infection like Candida. There can be viral infections like HSV, HSV6, BK virus, CME, adenovirus, Epstein-Barr virus, common respiratory viruses, RV, and late can be Aspergillus and other molds, late CME and varicella zoster. There are other complications like acute GVHD, immune suppression, and chronic GVHD which can kind of happen. Graft versus host disease can kind of happen along with this. So important thing is to remember that the first 100 days is the most crucial. There are a lot of infections can take place. CME is a big worry for us, resistant CME. Of course, other things are. So how can you get around this? So you can do prophylaxis by giving vaccine to the patient before that, but even that will be effective post-transplant. So one way of doing that is to supplement cytotoxic T lymphocytes, which is directed against these infectious organisms. Next one. And so this is the common viruses which can occur post-transplant. CME, EBV in the first 3-5 months, HSV first month, varicella zoster up to 1 year, HSV6 the first month, it causes encephalitis, HSV causes encephalitis, then CME can cause colitis, EBV can cause post-transplant lymphoproliferative disorder, and all these things are there. Pneumonia by respiratory viruses and adenovirus. Next one. So what you can do is you can collect the T cells from a donor, like you mentioned that universal T cell donor, or okay, and then or the donor of the patient herself. And then you can put, there are different ways of making it. The commonest and the easiest way is using a peptide mix. You collect the peptide from different viruses or fungi, and put that peptide mix into the T cell culture, and the T cells will react to that and then multiply. Then you collect this, concentrate these T cells, and surprisingly these T cells don't mount an immune response. They are actually directed towards those antigens only. They don't cause graft versus host disease. And you can use that during the first 3 months as prophylaxis. We are planning to manufacture this also. This is relatively easy compared to the CAR. You don't need a CAR T cell construct or anything. You can use that pep mix which is commercially available. This is another exciting area in immune, post-transplant infections, and very easy, potentially implementable in our system. Next one. 1 or 2 slides about the other. So there are NK cells which can be targeted like chimeric antigen receptor T cells. There is chimeric antigen receptor NK cell also. There are 2 types of NK cells, the CD16 prime which is cytotoxic, and CD16 dim which is immunoregulatory. A bit like Treg, that immunoregulatory, that is 56 prime and 16 dim. Next one. There are co-inhibitory and co-stimulatory molecules on the surface of NK cells also, like T cells. You can manipulate any of these to kind of achieve your target. Next one. And there is a say that this table mentions all those receptors. Next one. So here NK cells act when there is no MHC class expression. If MHC class is expressed, NK recognizes its self and leaves that cell. It spares that cell. Tumor cells, many of them don't express the MHC class one. So the NK cell will attack that. You can utilize this actually. They stress or damage body cell. No, otherwise normal cell may not express the MHC class, and therefore it may get attacked by the NK cell.
and the tumor cells with other antigens which can stimulate the NK cells will also cause lysis of that. Next one. So even if MHC class is expressed on the tumor cell, it could, that co-stimulatory ligands are that could stimulate. So you can utilize these properties to use NK cells. So bikes and trikes are nothing but, like I mentioned earlier, the bispecific antibody which will bind to the blinatumomab, which will bind to CD19 and the CD3 cells. There are antibody, bispecific and trispecific antibody which can bind to CD16 and an antigen in the tumor cell and connect up the NK cell. Just like connecting the T cells to the tumor, NK cells can be connected to the tumor. Next one. Next one. So the bispecific antibody which connects CD16 with the CD19, CD16 with CD19, and trispecific connecting 19 and 22 with 16. So 16 is expressed in the NK cell. So CD16 is in orbit like the T cell receptor. And you can use, you can also make a CAR NK cell, that's what here it is, that which will bind to the CD19, CD19 here. So either a CAR NK cell or bit like the blinatumomab kind of mode of action for NK cell. So that's another potential therapeutic target. So, but it has not taken off like CAR T cell because it's not, the effect is not that fantastic. Next one. A little bit about gamma delta T cell. Unlike whatever I talked about was a T cell receptor, was alpha beta T cell. There is a small subgroup of T cells called gamma delta T cells. They form part of the innate immunity. Only 1 to 5% of circulating lymphocytes. But when it comes to the tissue lymphocytes, they form a large area, especially of the surfaces, intestine and dermis. So it forms part of innate immune system. Means they are the foot soldiers, the infantrymen of the immune system. So it forms, wherever there is surface, it forms a large cell. Intestine, respiratory epithelium, and also the dermis in the skin. And they infiltrate solid tumors. So unlike alpha beta T cells, gamma delta T cells have the ability to infiltrate solid tumors. So these ones might be have more potential in solid tumors. Next one. So the same thing. Here again, co-stimulatory and inhibitory molecules are there. We can utilize that for activating the gamma delta T cell. Next one. One interesting pathway, all of you know about cholesterol synthesis pathway, and there is higher expression of HNG-CoA reductase in solid tumors. So, and zoledronic acid blocks this FFP synthase pathway. Why I mentioned this is, you can utilize this for activating the gamma delta T cells. Zoledronate activates gamma delta T cells. And if you infuse gamma delta T cells and give zoledronic acid, it can stimulate the gamma delta T cells and it will go and attack the solid tumors or other tumor cells which express higher amount of HNG-CoA reductase. That's one potential target. Next one. Again, the tumor microenvironment which can swing on to either side with gamma delta T cells. Next one. There are different types of gamma delta T cells, and it's more confusing. So there may be more to be discovered in the human side. Mouse, there are lots of different types of gamma delta T cells discovered. You could use any of these subsets for therapy. Next one. These are the different trials which are used with gamma delta. Mostly it's in phase 1 only. Next one. So advantage of gamma delta T cell is that it doesn't need MHC class, and it can bind the target cell without any MHC class antigen. It may not have an off-target effect. It will not have a mispaired T cell receptor like an alpha beta T cell, and therefore off-target effect is not there. Next one. With that, I'll mention one word about gene therapy. Gene therapy is also part of the entire milieu of cell therapies. And we happened to do the first gene therapy for hemophilia last month in the country. But anyway, the severity of the hemophilia is reduced from severe to moderate. But I suppose it's a working progress. Thank you.